Riscrivere il tempo. Invertire il corso dell'orologio biologico. Utopia o realtà? La prospettiva di raggiungere una terza e quarta età in piena forma e salute e di prolungare la vita ai 120 anni richiede comportamenti consapevoli fin da giovani. 70 tra i più autorevoli scienziati al mondo si riuniscono a Milano per parlare delle nuove sfide della longevità. Venite al Milan Longevity Summit dal 14 al 27 marzo a conoscere i grandi guru della longevità e imparare i segreti di una lunga vita in salute. L'evento è organizzato da Brain Circle Italia in collaborazione con le eccellenze milanesi e con il sostegno del Comune di Milano e della Regione Lombardia. Al Milan Longevity Summit si parla non solo di ricerca scientifica, ma anche di intelligenza artificiale, di lavoro e di organizzazione della società e delle città per fronteggiare il cambiamento demografico. Per consultare il programma, prenotare un posto o accedere alla diretta streaming, visita il sito milanlongevitysummit.org. L'ingresso è aperto a tutti e gratuito. Vi aspettiamo!
Welcome this morning. I'm very happy to greet all of you here. This is the beginning of our scientific days. We have a very long program. It lasts a week. We have discussions about politics, economy, what everybody can do to better his health and life. But these 2 days, I'm happy to say for the people here and also for the many people who are online with us that we have some of the most important and outstanding scientists in the world. And I thank everybody for having come to Milano. Milano would like to be a center of the future, a city where longevity is not only part of everyday life, but is also something which is planned for. Our mayor, who opened the works 2 days ago, was telling that he would like to make Milan a city where people can live long and in health. And our summit starts with 2 ideas. One is longevity, which means prolonging life. Some of it is natural. In the last 50 years, lifespan has become longer, of about 30 years. So, people are getting older. I think science tried to get them even older, but in good health. So it's not only important to speak about longevity, but also about healthy aging, which is something that maybe is less evident, but people are now, science is concentrating on this, because a long life is useless unless it is in good health. And I think I want to tell something about myself. In the Bible, it is written that God punished humanity. Humanity at the beginning was living very long. You all know Methuselah and all the prophets, they were living 800 years, 600 years. And at a certain point, God became upset with the human people because they were not behaving well. And so he said, I will punish you and I will shorten your life until 120. This shows how even in the Bible, the length of life was supposed to be the most important thing for humanity. And the punishment, the worst punishment God could give is to shorten the lifespan. Science now is speaking about living until 120, and not because it's inspired by the Bible, but because science is studying how our bodies naturally, unless we did bad things with them or bad things would happen, could live in good health till 120. So this is my wish for everybody who is here today and for everybody who listens to us online, to live a healthy life until 120 and maybe more. Okay? So this is to start, and I would like to call now, by the way, I am Viviana Kasam, if you don't know me. I am the president of Brain Circle Italia. It's a no-profit association. We Our goal, our mission is to spread science to everybody. All our events are for free. We count on the support of sponsors, benefactors, foundations, and I do want to thank all the people who believed in this conference and made it possible. I want to thank the speakers who are not paid to be here today, and I want to underline this: everybody came volunteer for the happiness to spread science and to talk about their very important research to everybody. This is not obvious, and this is why I'm saying it. I think it is very rare in our world today, who is run by economy and greed, that we have people who are ready to take long trips. We had somebody from Singapore yesterday, we have people from the States, and it's a long trip, it's hard, and really I appreciate what you are doing for us and for Milan. Okay, now I call Risa Starr. Risa is the president of the Academy for Health and Lifespan Research, and she's the director of the Longevity Biotech Association. Biotech, why biotech? Because many of the new discoveries need new technologies as well. So it is important to have research, and it is important how to apply research for people. And Risa, I would like you to tell the important role you have. You know, longevity is a very new subject. Till last year, 2 years ago, 3 years ago, we hardly heard about it. Okay, there were some books that came out, there were some scientists who became famous for their research, but a global approach to this in the whole world is also thanks to what your academy and association is doing.
Thank you, thank you. I'm so honored to be in the beautiful city of Milan, representing the Longevity Biotech Association and the Academy for Health and Lifespan Research. I want to tell you a little bit about each organization. So the Longevity Biotech Association is a group of about currently 30 longevity biotech companies and investors investing in the longevity biotech space. We were created to set standards in the industry. And we've started by publishing some white papers. One was about the definition of a longevity biotech company. And the reason that we did that paper is because there's a lot of noise in the field about supplements that encourage longevity. And the Longevity Biotech Association is really about setting standards to say what's real and what's, you know, we want to set standards for clinical trials. We want to set standards for what biomarkers should be collected. We have monthly events for our members where we talk about relevant issues. And we participate in the Copenhagen conference. Sorry, my phone keeps shutting off. And we are also about educating policymakers and the public and clinicians. So that's the Longevity Biotech Association. The Academy is a group of 60 of the world's leading geroscience scientists. Each year we have an election where the members nominate potential members and we vote on them. And we have a yearly conference where we collaborate on science and, you know, how we want to grow the organization. That organization is also about educating the public and collaborating. And we, gosh, sorry, I don't know how to keep this phone on. Both organizations have the long-term goal of acceptance of the concepts behind longevity and creating, extending people's health span. I think that that's the most important thing. As Viviana said, it's not important, so important. I mean, it's nice to live a long life, but you want to live a healthy life. So those are the organizations. I'd be happy to talk to any of you about them during breaks or during lunch. But we're really happy to be part of this incredible event. So thank you.
Okay. So we will start immediately with the first panel. What I want to say is that we try to make an interesting day. So we have different forms of lectures. We have tête-à-tête, which is two scientists talking to each other. We have panels and we have round tables. We don't have too many scientists today. I thought we would like to give time to each science rather than have a sequence of very long talks, and time also for the audience to make questions. We have people online. Luisa Capelli is in Rome now. She's collecting, driving, guiding the connection online, and so Luisa might send us some questions. So I hope this will be not only interesting but also enjoyable for all of you. And the other thing that we have in these 2 days is that I chose to have young chairs, okay? Because I thought that the voice of young people is very important. As we have heard in the last few days, longevity starts since when you are born. And it is very important to start knowing about healthy aging and what you should do to get to a healthy life, long life, since you are young. And so what we are doing this is not meant for elderly people. It is meant also for the young generation. And I know there are many young people here. There were many young people yesterday. There are many young people online. And I'm really happy about this. And now I would like to call our panelist, Nir Barzilai. Nir Barzilai is a great friend, a wonderful person, very generous, and he has a lot of emotional intelligence.
Flattery will get you nowhere with me, Viviana.
What?
Flattery will get you nowhere with me. I'm here.
Yeah. And he's the soul of the longevity science, I think. Nir is a director of the Institute of Aging Research at the Albert Einstein College of Medicine, and he is a scientific director of AFR. And he makes a lot of work to spread longevity and healthy aging science. When did you start this work of, spreading, organizing conference, going to the public, getting all the scientists together?
Oh, at the same day that God said that humans will be 120 years. It's exactly that. But by the way, God said 120 years, but it was at the time of Noah. Noah went on to live 960 years. And our fathers, right, Abraham was 175. There are plenty of people in the Bible that lived more than 120 years. So I'm challenging this concept. We might do better. Not today, but...
Okay. And then we have Joan Mannick. Joan Mannick. Joan, I don't know why, instead of giving me your title, they gave me your abstract. So this happens. But Joan is managing director and senior, no, this is Michael Ringel. So they didn't give me anything about you, which is terrible for a chair. And I have to scold my staff who gave me this morning. But Joan Mannick, I think you discovered rapamycin. Everybody, no. No, I'm wrong. You see, may I have a program, please, so I can introduce Joan correctly? Thank you. Sorry, this happens. Thank you, Joan. Okay, now it's better. Okay. You see, you are CEO of Tornado Therapeutics. That's right. Excuse me. Okay, so because I cannot introduce you properly, can you say something about yourself?
Sure. So I'm a physician scientist who started my career in academic medicine and then went to biotech, and I'm currently a CEO of a company making rapamycin derivatives to see if we can help healthy longevity.
So I was right about rapamycin. It's just that you didn't discover it.
Exactly.
Okay, but you all heard about rapamycin and the potential of rapamycin inhibitors, right? Okay. So, and the third speaker, and I have, thank God, your affiliation, is Michael Ringel, who came from Boston. Michael is Managing Director and Senior Partner of Boston Consulting Group, a very important group for economy, but you also are on the executive board of Sheba Longevity Center, and so longevity is one of your main interests. The title of this first talk is an introductory panel about debunking the longevity myth. And why? Because every day we read something about longevity, new discoveries, new promises, and you have clinics, and you have products, and you have supplements, and you have all kinds of things. But what is really effective? Is there something that is effective, that is proven by science to be effective? Yesterday somebody was saying it is very hard to have a long follow-up of the substances because you need 10, 15, and when you do this on aging people, it's even harder. So our talk now is debunking myth and saying what is the real science, what we should follow, and maybe it's not yet gone on trial completely, but gives good hopes on human, what doesn't, what is the economy. I like to say I'm a baby boomer, okay? Baby boomers are a very, very special category of people because we are the most numerous generation in the world. We lived under the economic boom, so many of us were able to risparmiare, how do you say that in English? To save money, so we have some money. We usually don't have any more children to send to school and to pay for. Many of us, unfortunately, don't have our parents anymore. So here we are, so many, with money, with some money, and guess what? We are a great target for marketing. And what a person 70-year-old or older has at heart, most of anything, living healthier, maybe living longer, and enjoying life for whatever time there is. So, of course, the market knows this. And as a baby boomer, we have been at the center of market interest for a long time, but now especially. If you read newspapers, books, products, cleaning, cosmetics, clinics, everybody promises anti-aging, healthy aging, better memory, better performance, and everything that is connected. So, Nir, I'll start with you. What is true in these promises, what we can hope for, and what we should beware of?
But by the way, we had a conversation before, what are we debunking? So thank you for clarifying some of the things. But like we're debunking we're not immortalists, right? Let's start from this, and we're not immortalists. We're not fighting death, we're fighting aging the way it is. And you're right, from our perspective, I guess, or from my perspective, there are two environments. We are two antagonist movement. One we call the anti-aging. Okay, those are the people that you kind of said they have supplements, they have other things. They tell you that there's a promise for what they're selling. They tell you that you'd live forever, and if you die, nobody sues you. Okay, and we know little about the supplements. We had exception yesterday of chocolate, right? Chocolate actually has clinical data for that. We are the other side. We call ourselves geroscientists. The people who you invited here are geroscientists. A lot of them are in the academy that Risa mentioned. And we are trying to be responsible people. You called the day the revolution of aging. Revolution no revolution has ended well or not bloodied. You know, I don't want to be a revolution leader, but we're on the opposite. We're trying to be very responsible. We're trying to, you know, to debunk the myth and to say what we know in a very conservative way. And so we believe that aging is flexible. We believe that there are things we can do about it. We know how to proceed, but we're not selling snake oil and we're not making promises where there's only hope. I'll end with that.
Joan, you are both a scientist, but also you're involved in a company, okay? So you have the two sides, science and applied science. So what is your understanding and opinion of the field?
So I agree with Nir. There is no doubt in my mind that we can develop medicines that will extend healthy lifespan in humans. Absolutely, because there's so much data in preclinical species this is possible. But to prove that any particular medicine or supplement actually has health benefits costs millions and millions and millions of dollars. And this work needs to be done and needs to be funded, but the work hasn't been done yet or reached a level where we know for sure that there is any medicine or supplement other than the one supplement Nir mentioned, which is an extract of chocolate, not just chocolate itself. That's the only really convincing data that any supplement will have major effects on health. And there's some data that a vitamin, multivitamin, may have benefits for cognitive function. But other than that, everything that you hear on TV where they're saying, take this supplement and you will have better cognitive function, you will have better energy, there's no convincing data yet for any of them. And it's so important that the public realizes that just because they say on TV or in an ad that these supplements or stem cells or whatever it is are going to help you doesn't mean it's actually true or there's data. And so I think right now the things that there's data for is exercise, sleep, you know, probably having a balanced diet with fruits and vegetables, and that's about it. But those are important. You know, there are things we can do right now, and everything else we just need to put in the money to figure out, do the placebo-controlled trials to prove what actually works and what doesn't, because I guarantee you some of the things that are working in preclinical species will work in humans.
Yeah, and also I think that some things will work on some people and not on other people. Right. For example, today we will have a great scientist and also a person who animates debates on this, Antonella Santuccione. She will talk about why women live longer and age worse. And this is something we don't know, but one of the things which is important is to have trials on men and women and on different categories of people. There is, yesterday we talked about a lot of comorbidity. So there are some things that are good if you are in a health state, or you know you have some kind of problems, but might not be good for other things. So I think that there is a great need to clarify, you know, and we all have illusions. I mean, I every day would like to buy some magic cream, some magic supplement. Of course, you know, I want to look better and to live better. And when my memory fades a little bit, I'm worried. I said, maybe I get Alzheimer. Let's get the magic pill. I think this happened with most of us, but it happens also with young people. I know a lot of young people who are taking a lot of supplements. And of course, you have the example of millionaires who are taking, and they go online and they say, I take 50 pills a day and 60 pills a day, and look how great I look. So, you know, I think illusion is part of our dreams. And what about you? You are in the business, in the economy field. What do you think of this market of longevity?
Yeah, thanks for having me. So panels are no fun when everyone agrees. So I told Nir and Joan, no matter what they said, I was going to take the opposite side. So it's good you positioned me across.
I took the opposite side, by the way.
You can do the opposite side of yourself, I think, Nir, but that way they can't grab the mic out of my hand. When you look, there's demographic studies done where they look across the population of all the different countries, and they project out what's the longest possible lifespan we could get. Excruciatingly detailed, very accurate work saying you can't get past 120. But they asked the wrong question. It would be like inventorying the highest altitude that humans had achieved in the 1500s, and you checked all these different populations and found, well, most people live at about 1,000 feet in altitude, and no one's gotten above 29,000 feet, which is correct, right? You can't climb higher on land than that. But if you have new technology, it is actually possible to exceed that altitude, and I just did it a few hours ago flying in, right? And that's the promise, that there is the possibility that we could extend lifespan well beyond that number. We know that's possible in animal models. So you can take a worm and with a single gene mutation extend its lifespan 50%. There are, deeper interventions you can even get, the record now I think is now approaching like 10 times extended of lifespan. Now, these animal models are not humans. There's a lot of reason to believe that what's called R-selected species that have very short lifespans, it's going to be easier to extend their lifespan than a human lifespan. We have no good data that you could do an intervention like this in humans. If you walk down the sort of hierarchy of what's good data, as you go further down that hierarchy, I think we get some hints that it might be possible. So the absolute gold standard data is you have to run a randomized clinical trial. You do an intervention in a set of people, you do a control in another set of people, you see whether there's an outcome. Now, lifespan trials take a long time in humans, so seeing that would it's prohibitively difficult right now to do that. The next layer down are a combination of various other ways to make inferences. So epidemiological data. So if you look at people who exercise more, do they live longer? If you look at people who eat a plant-based Mediterranean diet, do they live longer? You see these associations. Now, epidemiological data is fraught with risk because the causality is never clear. There are confounders. But where you have some additional data to say, okay, I can do let's say a small trial and see some of the biomarkers move in the same direction experimentally that I saw in the epidemiological data, it gives you some more confidence around it. Then there's preclinical data, right? If I can do an intervention like rapamycin, which was the first intervention proven to extend the lifespan in mammals in 2009. There's something called the National Institute of Aging in the US. They run something called the Interventions Testing Program, ITP. They test a bunch of different agents. Lots of things have failed, but they have shown that it's possible to extend maximum lifespan in mice. Something like, I think rapamycin was like 9-10%, that sort of range, right? Those data don't necessarily translate to humans, but they give you a clue that maybe there's something there. And then you have some additional data when you understand mechanisms. That's sort of a next layer of data. You can say, well, is there plausibility to believe why that would be the case? And Joan, I'm sure, can talk about, mTOR is this important switch that exists in metabolism across different animals, including humans, that switch you from sort of growth and reproduction to repair and maintenance. And the repair and maintenance state is associated with living longer. And so there is a mechanistic reason to believe where it might be true. So we have 0% of the gold standard data in humans yet. But we have other kinds of data that give us at least an inference that it may be possible. And that's why this field is so exciting, because we have to do the work now to actually, do the science. Is it actually true or not? So right now, most of the supplements, not a lot of data behind them. You may be even taking a risk. But do I believe that we'll get there someday? Actually, yes, because there's a lot of layers of evidence that make me believe there's something there.
And then there is something I think great about our brain, that sometimes things help because we believe they help. And so, you know, I would like to say that when you take something and you think it works, it might work, even if there is no science. Maybe it works for science, but it's not proven, but maybe it also works because you believe it does. You know, when I started my career as a journalist, I've been a scientific journalist, we were talking about placebo effect. And people were saying, placebo, no good. You know, if it's only placebo, it's it's no good. It's something you imagine, but it doesn't help. Well, I think that placebo is the best possible medicine because you get cured by your brain without taking any substance which might have a bad effect. So
I love that, Viviana. So all the trials that I have done are always placebo-controlled. And it is remarkable the benefits that people have getting placebo. So they're told you're either getting a rapamycin-like drug, which extends lifespan in preclinical species, or you're getting placebo and they don't know what they're getting. And so they think, I might be getting something that could extend my lifespan. They have more energy, their golf game gets better, they have like better sexual function. It goes on and on, the placebo. And you're right, the placebo has no health risk. The problem with every single therapy that actually has a biologic effect is if you have a biologic effect, you will have a side effect. And that's why you have to do these trials, because if you're going to put someone at risk of that side effect, you need to have a benefit that outweighs that safety risk.
But in the meantime, everybody can take a placebo outside. They have the placebo, and you'll see that you'll feel much better.
We're starting a company called Placebo Incorporated.
Right. I'm trying to be antagonistic to you, but I agreed with everything you did. But I want to say something, and I'll show data tomorrow. There are 12 FDA-approved drugs that when you give to animals, they live longer and healthier, okay? And we published papers. The last one came actually last week. And of them, there are 4 medicine that gets, it's a 12-point scale that get 10 to 12. And for example, I'll tell you all the medicines because it's important, because things are happening now. And any doctor can repurpose any drug. There's a diabetes drug that is of a class that's called SGLT2 inhibitors. When you give it to animals, they live longer, they live healthier. It targets a lot of the hallmarks of aging. It's designed for diabetes, but it has effect on kidneys, it has effect on heart. There's abstract on cognition, but we don't like abstracts for now. It has to be published. And it decreases overall mortality by like 25%. Metformin is the second. We'll probably talk about it. The third is biphosphonates. Okay, it's the osteoporosis drugs. And the first time I heard about it, I read a paper where women on biphosphonate don't die in ICU. Okay, but then you give it to animals, they live longer. And in clinical studies, controlled studies, there's decrease in overall mortality. And the third, guess what, is the GLP-1, the Ozempics, okay? They also showed they were developed for diabetes, but they're good for obesity, and in obese people, they also decrease cardiovascular disease and overall mortality. In other words, there are drugs available now. How to use it, we don't know, but those are drugs that have gone, okay, so the advantage of drugs over supplements is that drugs that are FDA approved, they're safe, okay, they're efficacious, you have data about them, okay, so you don't have to start from start. And a lot of drugs, I'll take metformin for example. Metformin has been used by doctors for obesity. It's not a good drug for obesity. You decrease, you have 5 pounds, but you need more to lose usually. It's repurposed for PCOS in women. It's repurposed as pre-diabetes. There's a DPP control trial that showed that it decreased risk of diabetes. And now we know that people who get COVID within the first day, if they get metformin, it decreases hospitalization, death, and long COVID by 50%. Okay, so people can prescribe any of those drugs that have already shown to decrease overall mortality.
So maybe if I jump in on this. So I'll take your first on the list. So canagliflozin, which is the SGLT2 inhibitor that worked in the ITP, and probably others in the class would do the same. They just didn't test the others in the class. So there's a class of drugs that reduces the ability to reuptake glucose out of the urine in the kidney, so you end up excreting glucose. No one should take that without advice of a physician. Every drug on the market has a risk-benefit. There are risks. You can get diabetic ketoacidosis, right? There are risks to any drug on that list. But usually drugs are approved when the benefits outweigh the risk for a set of people. When you look at the data on canagliflozin, it hits the sort of hierarchy of data that I described. So there's randomized controlled trials showing a mortality benefit in diabetics, not in non-diabetic patients. There are data in the animal models that we just described in the ITP, and there's mechanistic reasons to believe this, right? So the best interventions that are the longest proven in multiple species are things that hit glucose monitoring, nutrient sensing. Frankly, the best thing, the most validated intervention is caloric restriction, right? Just don't eat so much. And they're all hitting this pathway around glucose sensing. Even mTOR, frankly, is downstream of that, and autophagy further downstream of that. So there's a mechanistic reason to believe in this class. So it hits multiple layers of evidence, which gives you some confidence in it. At the same time, it has not yet been shown in non-diabetic patients to have...
It has, by the way.
Oh, it has? Okay.
Yeah, it has a non-diabetic patient.
Yeah.
Decrease cardiac and renal outcome and decrease overall mortality in non-diabetes.
Okay.
All the last New England Journal are non-diabetic.
You want to say something?
So I would say we're not talking... If we're talking about extending health span, not treating renal or cardiac or diabetes, and you go into a healthy aging population, we just don't have data yet. And it's not, I agree, it's not that there's not good promise, but we don't know. And you take Ozempic. Okay, Ozempic loses muscle mass. You may not want to take an elderly person and have them lose more muscle. So we have to do the trials in the population where we actually want to go in the geroscience field and prove these drugs work. Because right now we don't have that data.
Yeah, and I didn't mean, I think I said at the end, we're not ready, we don't know when to use them. And there are trade-offs for everyone. But I want to turn it to positive, because this Ozempic thing where you lose weight, you lose lean body mass, okay? But then you carry less weight. So, it's usually okay. But not in elderly people. Elderly people, sometimes they lose this lean body mass, and it's actually, even they have less weight, it's a problem for them. So there comes geroscience, and there's several ways. One of them, a company by the name of BioAge, they are part of the LBA. They have this peptide apelin that they were going to use for sarcopenia, but now they have a thing with Lilly, $270 million, where they're going to show that co-infusing of GLP-1 with apelin will preserve the muscle and you'll lose only fat. So that's where geroscience comes to help even the development of drugs now.
I would like to ask you, Nir, for people who are lay people, and we have many listening to us, why is lowering the sugar good for living longer or for living healthier?
First of all, for diabetes, lowering sugar is good, okay? We're actually talking about drugs that hit the hallmarks of aging. And you'll hear a lot about it today, but the nice thing about those hallmarks of aging, you target one, you target the other also. You know, a lot of those drugs, as I said, a lot of those drugs, rapamycin and metformin, they hit all the hallmarks of aging. It's true that some of the drugs came because of diabetes, but that's not how it started. You know, metformin for 30 years was prescribed for arthritis, to prevent flu, for other things when they noticed that it lowers glucose, okay? But a lot of those drugs, in my mind, yeah, lowering glucose, but you know, they never lower glucose. When you take this drug and you don't have diabetes, it doesn't lower glucose. It lowers insulin. It doesn't lower glucose, okay? So it's not the glucose sensing, but it's not only the metabolism. It's the autophagy, you'll hear more. It's about many other things that are happening in the body.
What about, we heard a lot, everybody heard about rapamycin. What is the effect of rapamycin? Why do you think it might help for aging?
I think one of the reasons is, so rapamycin is a drug that in every species studied to date, yeast, worms, flies, and mice, extends lifespan. And they span 1 billion years of evolution, so it's really likely that rapamycin will have some benefits in humans. But rapamycin and its derivatives that are called rapalogs also have side effects because you are, again, targeting biology that's fundamental. But what happens as we get older in some of our organs is this mTOR protein that rapamycin targets gets hyperactive. So I think just turning the protein down to younger levels may be one of the reasons why it has these health benefits. And as we start understanding these signaling pathways that get off as we get older, it's like retuning a car. If we just retune them back to a young level, that may be what is responsible for health benefits.
Thank you. Would you like to say something, or we go to the public?
Let's go to the public. I do have a thought,
Okay.
but I
Are
think
there
it's
any
probably too
questions?
orthogonal.
Yeah, there. Please, can you bring the mic? No, it's okay.
We can hear you.
And also, I'm wondering, Luisa, you are online. Are there any questions online? You have. You'll ask them, Adele. Okay.
Dr. Barzilai had said that about FDA drugs being safe, but I know that over 10,000 have been recalled in the past couple of decades. Do you have a comment?
You're right, but no supplements have been recalled, and they're probably causing lots of harms too.
Nir, didn't you mean, I think you meant the safety is known, like what are the risks versus the benefits for the FDA-approved drugs, whereas the supplements, nobody is required to look at that.
Yeah,
right.
Maybe I'll comment on that. So it's, I think, about 3% of drugs that get recalled. Medicines go through an enormous amount of testing, right? In clinical trials, you may have thousands of patients where you're looking at the efficacy and you're looking at the safety profile. You get only approved if that is convincing to a body of scientists that there's a statistically significant signal. You generally need 2 trials that show proof of efficacy as well as safety. So there's a lot of rigor that goes into them. However, you know, the sample set there of a couple of thousand people is different from a drug that might be, once it's on the market, in hundreds of thousands of people. And so regulators, FDA, EMA, usually have a requirement for a monitoring program, and the sponsors, the pharmaceutical companies, usually, you know, even if not required, will do a monitoring program to look at signals. And when a signal is detected, maybe it's a small change in risk that you wouldn't detect in a 2,000 person trial. It's just not enough sample size to detect it. But in several 100,000, you could. When a signal is detected, it's a great sign that the industry and the regulators then recall drugs. And that's the difference with supplements and other things that are not tested, and there's no proof and there's no ongoing data against them. You're actually more at risk than in a field where there's this level of rigor in the assessment.
Yes. No, we need the mic.
Here.
There's somebody with mic there.
Yeah. So rapamycin is being used as an off-label drug in the U.S. and around the world, and I wanted to know your thoughts on the protocol and cycling rapamycin every 2 months or 3 months.
So a lot of the rapamycin dosing was based on a paper that I actually was involved with in 2014, where we dosed 50 people for 6 weeks with two different doses, 50 people at each dose, and there was an improvement in vaccination response. This is a really small number of people. In those 50 people, we saw the better vaccination response and no side effects, but as Michael was just saying, as we dose more people, we'll get a better sense, is that safe? What are the side effects as you extend the dosing beyond 6 weeks? What are the benefits? Who's benefiting and who isn't? And we have none of that data yet. So I would say it's still too early to be taking rapamycin because we don't understand all of this data. And I think we can get the data with rapamycin. You know, we at Tornado have a different kind of rapalog we think might be safer, but we just need to get the data so that we can be smart and tell people as physicians, this is, the risk benefit of this is good for you, and we aren't there yet.
And maybe I'll jump in on some of the preclinical data around this. So mTOR occurs in 2 complexes, mTOR complex 1 and mTOR complex 2. You know, super insightful naming convention there. Rapamycin binds FKBP12, which only will interfere and bind with mTOR complex 1. So there's a belief, not fully validated, that the benefits are mediated by complex 1. The downsides, which you can get hyperglycemia, you can get dyslipidemia, right? The downsides more mediated by mTOR complex 2, which has different pharmacokinetics because it's not directly bound. There's a belief that if you pulse dose, you're hitting only complex 1, less complex 2. There's also some preclinical data to support that. Ariel Apelo wrote a paper out of Dudley Laming's lab where they did it in mice and they did pulse dosing and saw, a change in the risk-benefit profile. Too early for prime time, as Joan would say, but that's the thinking behind why folks say, okay, pulse dosing as opposed to, constant dosing.
But can I say something more general that I think is important because we have young people here. And most of the studies that are being done that are relevant to us and relevant to age-related disease are recruiting over the age of 50. A lot of them, the average age is 60 and 70. A lot of those drugs are not good for young people, okay? There is an antagonistic pleiotrophy hypothesis of aging. Not everything that's good for young is good for old. There's a lot of examples like that. And also, not every medicine that's good for old is good for young. If you give rapamycin to young exercising person who don't have increased mTOR, you'll decrease their ability of their muscle to grow. Metformin actually has also an mTOR inhibitor, but metformin, for example, decrease growth hormone, IGF-1 growth hormone, which is good in old, but not good in young. Decrease testosterone, maybe it's good in old, not good in young. So I want to just warn, we're starting the breakdown of aging. We go, and then we grow, and then we start breaking down. And so the treatment for this part of aging is very different than the mechanism of aging until we get there.
Thank you. There is more questions here?
You listen to me? Yes, Alberto. FDA does not recognize aging as a disease, right? So the likelihood that one day we will have an anti-aging pill licensed by FDA are almost nil. So essentially we are talking about drugs that will be licensed for treating diseases and eventually used off-label as an anti-aging pill, let's go, as prevention, okay? Do you think this is a likelihood scenario, or do you think there is a possibility that one day FDA will approve a drug as an anti-aging pill?
So I want to get to this because I disagree with the statement, which we often are saying in our field, that the FDA does not consider aging a disease. all the FDA wants is an endpoint where you show that the risk is outweighed by the benefit. So they just say, what is aging? What are the benefits that you are going to show and define as aging? So Nir has a trial he'll talk about to define what does it mean what is aging. And when I was at Novartis, we had a big discussion with all the heads of the FDA divisions, and they said, you know what we think would make sense is target with rapamycin or derivative. First, see if you can make the heart better or the immune system better or the muscle better. And if there's a dose where you can show you make multiple aging organ systems better, we have no problem giving you an anti-aging indication. So I think they just need us as a field to define what that is going to look like in terms of an endpoint. It's not that they aren't going to let us have that kind of indication.
So we have a study that will launch this year. It's called Tête. And the only reason we're doing this study is exactly what you said, is to show the FDA that we can target aging, right, the way we think. And we discuss with the FDA this study, and you can see it in a Howard, in a film that's called The Age of Aging, and they came to the FDA with us. And the point is that we are defining a cluster of age-related disease, cardiovascular disease, cognitive, cancer, and mortality as a cluster that we're going to prevent or move. And if we do that, and that's what we discussed with the FDA, we don't want to get there and you say, oh, you should have done a different study. The FDA said, yeah, if you are going to show that you've done that, then this is going to be a good indication, moving a cluster of age-related disease, and this study will be the template for the industry.
I'm sorry, just another additional comment. But that implies that you must have highly well-defined biomarkers that will be, validated enough to convince FDA that you have, or not?
No, the FDA doesn't want biomarkers for this study. It's the opposite. This study will help us to establish biomarkers. But I have to tell you that in the meantime, at the American Federation for Aging Research, we have an initiative where we're taking studies that were done on metformin, on SGLT2, on GLP-1, and we want to see how biomarkers change within a year. The important thing about biomarkers is not only that they tell you your biological and not your chronological age, but that they change with the intervention. So we're going to see what biomarkers change, omics biomarker, lots of them, what change with the interventions, and then how common are the biomarkers that change the rate of aging, basically.
Okay.
I have a question. Some of the scientists say that aging is a disease and can be prevented and cured and must be considered as a disease. Is that true? Does it have any basis? Because I think that aging is natural. I mean, animals age, people who don't have any bad things in their life still age. So is it serious to consider aging a disease that must be cured?
My answer to that is that aging is the mother of diseases. Okay, aging drives diseases, but I'm against calling aging a disease, and I'll tell you why. The elderly, look what happened in COVID. We took the elderly and put them in islands, isolated them. They don't want to be called sick. What, you're 60 years old and now you're sick? They don't want to be sick. American Federation of Aging Research doesn't want to call them sick. FDA doesn't want to call them sick. And we found a way to do a study where we don't have to name names, okay? We just have to show outcomes. And it'll be clear. By the way, the FDA didn't agree with us that if we show that we prevented aging. They said, no, if you prevented a cluster of age-related diseases, that's fine. I think eventually it will become like a thing, but we don't need it now.
So I think it's a risk factor for disease. It's not a disease. It's great. If you have the opportunity to age, you are doing well. You're continuing to be alive. And if you stay healthy, you're still aging, but you're having a great life. So it's just a risk factor that increases the chance you won't be healthy, and we can target that risk factor.
Well, so I have to take a different point of view. It's a definitional question. Like it's sort of a nonsense question. So if you define a disease as something that only happens to a subset of the cohort, then of course it's not a disease because we all age and get infirmity from it. If you define disease as something that causes infirmity, then it is, right? So it's sort of a level of, not directly, but indirectly causes infirmity. And so it's a definitional question of what you want to use is, well, what is a disease?
Okay, thank you. Are there a question there from the...
I have a question from, we received a question from an audience, outside audience. It's about the vitamin D and supplements in general, okay? So I add on that. Vitamin D has been dismissed based on some data about showing that it does not prevent bone fractures in elderly, right? But at the same time, there was several evidence that people with very low levels of vitamin D have a much higher chance to develop bad COVID, severe COVID, okay? So when we say supplements are not useful, maybe we should just dig into and specify which supplements. You see what I mean? You agree with me?
Right, I think what you just have to show, which they're doing with vitamin D, is there data that a vitamin helps or not, or doesn't help? So for instance, vitamin E, natural thing, it's an antioxidant, cardiovascular disease has too much oxidative stress. Everyone thought vitamin E would help your heart. And when they actually did the study, it doesn't help heart. You just get as many heart events when you take vitamin E as if you don't, and in fact, there may be some evidence you get more heart failure. So until we get the data to understand what populations we should use these supplements, we shouldn't be saying anything, not that we can't get the data to prove when they will work and when they won't.
And you said vitamin E or D?
D, vitamin D.
D.
So
D.
So the amazing thing about vitamin D is that it seems to be risk for many diseases. And there are many clinical studies that gave vitamin D and they helped nothing. Okay, that's the state of the art of vitamin D. Bigger studies, clinical studies, and they helped nothing except people with osteoporosis. Okay, so this is an example of not every marker that we have that is associated with our disease is also going to fix this disease. I mean, it's real that people with vitamin D have that, but that doesn't mean that vitamin D is helping. With cholesterol, we know if your cholesterol is high and you lower that, it has effects. Okay, but with vitamin D, we don't have this evidence yet, except osteoporosis.
Are there more questions, or you want to say something?
Just one sentence on it. So if the inference you took away was medicine's good, supplements bad, that's not the inference. I think the inference we were trying to get across is evidence-based medicine. Make sure you have clinical trial data to support your claim.
Okay, thank you very much. I have a question about the NMA and PQQ, which are very popular in the market now. I would like to know if it has a relation between the longevity and the anti-aging. How do you see those ingredients?
NMN, you said?
NMA. NMN.
NMN.
Yes, and PQQ.
And what was
PQQ?
PQQ.
Yeah.
So, go ahead.
Look, NMN, you know, in my lab, when you increase NAD in animals, it works. Mm-hmm. In humans, we don't have data for that. We just don't have data. And I'll tell you, I'm reviewing a lot of papers, and I'm seeing lots of signal, but there's no single evidence that NMN will increase what we try to increase, which is NAD, which is the energy sensor of the muscle. We really don't have evidence on that on humans. So I'm not taking any. That's all I can say.
But you're taking metformin.
Pardon? I'm taking metformin.
Yeah, he's taking metformin. Just to give you an idea, I'm not taking it. He tells me I should take it.
I didn't tell you you should take it.
You said, no, okay, sorry.
You think I'm saying that.
I'm thinking you're saying that, but what I'm saying is that I am cautious about metformin even because the quantity of metformin you have to assume to have, probable benefits is rather high. And I say, well, I'm well, why should I take something that is not proven and that might be good for me, but I don't know. I'm still well, so I'll try to manage without metformin.
Viviana, you're doing so well. I have no advice for you. You give me advice now.
Thank you. Okay.
I have another question. Yeah, I'm sorry. I read some articles which mentions that the most of our disease and aging problem are because of the inflammation. And how do you see this point? Yeah, thank you.
So, as we get older, there's sort of an increase in what's called sterile inflammation. Usually you have inflammation because you have a wound or you're infected, but just at baseline, there's an increased inflammation. And yes, there are health benefits in preclinical species when you decrease that inflammation, and some of it comes from senescent cells, and Laura will be talking about that maybe today or tomorrow. But we, again, don't have the data yet in humans that any anti-inflammatory drug or an anti-senescence drug has health benefits. It's not that we won't get there, but right now we don't know a dose, a duration, the particular drug where the benefits outweigh the risks. And I guess you were mentioning PQQ. Is that a mitochondrial targeted drug? Again, just no data in humans for any of this yet that is, you usually need thousands of people in a trial to have it powered to be sure you're seeing a clinical benefit, and we just don't have that data.
Okay, I'll allow one more question there, and then okay, 3, but fast, okay? Make fast questions.
One answer each.
I don't want to run over time.
As you know, we know many mechanisms involved in the aging process. How can you address with just one single molecule one of the thousands or hundreds of mechanisms that are involved in aging? That drives me crazy.
Well, but isn't
Why
that
do you
great?
take meth for me?
Isn't that great? So, look, you'll hear more about those hallmarks of aging, but the important thing about them, they're interconnected. You fix one, and, Laura, Anna Maria later will show you. You can fix autophagy, you treat inflammation too. Am I right? Right? So the nice thing is that that's what happened. Now, think of it this way, okay? You have drugs that take your old cell or old organ or old body and make it younger. It fixes lots of things, okay? That's what happened. What does it do first? We don't know yet, okay? We're trying to understand. But the truth is that this is such a breakthrough for us because we know that any target will affect the other, and that also means when we're developing drugs that maybe it does something in inflammation, but it will do metabolism too. So maybe it can be an anti-diabetic drug, right? I mean, you have many possibilities also of drug development by that. But just think about simple. Those drugs are doing something to aging. We might not understand the exact mechanism. And the hallmarks are not the mechanism. They are drivers, but they're not necessarily the mechanism. But you fix them, and you get all those benefits. Don't go crazy.
But I think maybe another way to think about it, the signaling pathways are interconnected. So intervening in one tends to change the signaling in the others, so you get more than one downstream output that impacts aging because of that interconnectivity.
I would rather go to mitochondrial and work on them eventually, because that is more basic, probably. It's another way of working. It's another way around. Maybe Luigi.
Luigi, right. Well,
The nice thing is everybody has his favorite hallmark, and they said, I would concentrate on that.
That's the reason why you drive me crazy if you just take one.
But what I want to say is, now, you hear a lot of doubts here, but the great thing I think we have to underline is the first time in history we have proofs that something can be done, and that there are systems, not necessarily drug, but also CRISPR and cell works on cell and epigenetic that can reverse aging. Maybe it's not for our generation, unfortunately. Maybe we will find out that it's too dangerous and we cannot do it. But I think that for the first time in history, there is a sign that something can be done, that aging is not necessarily means the worst part of your life where you're full of diseases, and that maybe we can, in time, get younger and avoid aging, which I think is the reason we are making this summit, to tell you what is the future. We don't know yet what will work or not, and this is the round table. And I really wanted to start this because you will hear in the next few hours and tomorrow fantastic things we could never think about, dreams, for the whole of humanity. Sorry, my English. I try to do my best. But I want to warn you that what will be said here is not proved yet, doesn't have enough trials. Sometimes it works on animals and doesn't work on people. Sometimes it might work on men and not on women. And so, this is an insight into the future, but we have to be very careful now. Please, you wanted to say something?
I think you wrapped it up perfectly.
Yeah, that was such a perfect summary.
Wow, I got a degree. Okay, time for a couple more questions. Okay, there. Microphone is coming. 2 more questions, okay?
Very quick question. Actually, it's a comment. I don't know exactly what Dr. Barzilai study will be, of course, but I was struck by the thought that metformin, GLP-1 inhibitors, and sodium glucose cotransporter, so agonist and cotransporter inhibitors are currently 3 drugs that are taken by most people with type 2 diabetes. By now, they all started on metformin. Little by little, they added the cotransporter inhibitors, and now they are adding the GLP-1 agonist. So in a sense, you're going to have, within a few years, very soon, a large population of people who are at a disadvantage with regard to lifespan, because diabetes has been known, even type 2 diabetes has been known for many years to decrease lifespan. And however, taking 3 drugs that might have some effect. So I was just wondering if there is on the kind of people are thinking of at least an observational type study over 10 years to see if people who are at a disadvantage in comparison to a cohort who has like Mrs. Kasam doesn't want to take any drug because doesn't need such drugs, or like me, and compare if there is some advantage over certain, even just survival.
Terrific. The answer is no. There's no such studies, but they're drug studies. And then if you're asking about what happens in 10 years, we're not there, not with the GLP-1, not with the SGLT2 now, so it'll come out. But I do want to say something, and maybe it's a question for you. If we have several drugs, metformin also is an mTOR inhibitor. Say people are on mTOR and on your drug and maybe some other drug, aren't we inhibiting mTOR too much? Is there, are we also having medications that we'll have to consider their antagonism or their bad side effects?
Yeah, and I would say it's clear that these drugs are good for diabetics. But are they good for someone who doesn't have diabetes? I don't think
yes,
we'll
the
know.
answer is yes.
But Or heart, like there are specific populations that all these studies are done in that may not be generalizable, or we just don't know yet, to people who don't have certain underlying comorbidities.
Well, is rapamycin good for people who have diabetes?
Probably only if you use a dose that doesn't inhibit TORC2.
No, the point is that rapamycin, people are saying that rapamycin is a drug that is not good for glucose metabolism, basically. They're not not the one that you're developing, rapamycin.
What low dose rapamycin could be, we don't know. But I would say your point is correct. When you combine things, we don't think you want to inhibit mTOR totally. You want to bring it down to young levels, so too much isn't going to be good.
Okay, I want to say something and then we have to close. There are people maybe who are not English speaking. I think many of you have a simultaneous translation. If you want to make questions, you can make them in Italian. You don't have to make them in English, because for some people it might be difficult to make questions in English. So feel free to use both languages. We have great simultaneous translator. I thank Marta Bocchicchio, who works with me and who is in charge not only of the organization but also for the translation. And so I hope everybody can enjoy it. I would like to give you the last word, because you spoke less than everybody else, so you need some extra time. But I want to ask you, in all this global investment that is doing, I call it the gold rush. It's like in California, the beginning of last century, everybody was running to find the gold. Is there some gold? And where?
So I'll talk about it more at length tomorrow. So look, there are two countervailing forces. There is the natural desire of humans that want something to believe, overhyped claims and charlatanism that comes with that. And there's a real risk in this area that you have folks making claims about things. Either they actually believe it, or maybe they don't, but those claims are not founded. So that's the risk of the rush we have right now. Fundamentally, there's a tremendous amount of economic value, and I'll talk about it tomorrow, if we could extend health span and or lifespan, right? People are productive longer, they're able to be part of the workforce longer, enjoy the world longer, do things that benefit the economy longer, which, maybe that's the least of the considerations, but it still matters. And they're drawing down less in terms of resources for things like entitlements, medical payments and so on. So it's absolutely, I'm not going to steal the thunder from tomorrow, but it's absolutely astounding how much value there is there in even small changes to lifespan at a population level.
And the health span especially.
Correct. And if you are able to come up with medicines or other interventions that actually unlock that, you should absolutely accrue some of those. Some of that goes to the manufacturers and discoverers of that, and some of that goes to society. And the numbers are absolutely off the scale here. So it's worth us doing. It's also, it's the search for the philosopher's stone, right? It's like the thing that captures the imagination, I think, of human history more than any other, thing out there, at least for me. And we're at the moment where we can at least start to make some progress on it, and that's why I'm particularly excited by it.
Thank you. So if you have more questions, our speakers will have their time of talk today and tomorrow, so you can listen to them in a... Yeah? Sì, ma veloce perché...
È questa: quando io ho chiesto ad un ordinario di endocrinologia per quanto riguarda la metformina, c'è stata una forte resistenza, molti dubbi si sono stati espressi. Quando ho parlato con un nefrologo ha detto: attenzione che noi abbiamo grossi problemi per quanto riguarda l'insufficienza renale. Come possiamo superare queste resistenze, professor Barzilai?
I agree with you.
No, he said that normal doctors, when he asked to use certain, metformin, they oppose it. And I think that one of the things I can say, and then you'll answer with a more scientific point of view, is that usually doctors are not prepared normal doctors to do something about aging. It's a new science, especially in Italy. You have a lot of gerontologists who take people in care when they have some sickness. There is not a preparation, and this is something we are trying to do with our summit, is to make public opinion conscious that there is also a new knowledge and a new preparation that must be given to the medical class. Because up to now, we had only, you go to the doctor when you're sick, when you start having Alzheimer or Parkinson or osteoporosis or whatever, and maybe it's too late. And I think the doctor should learn how to take people in care when they are younger and explain to them how can then prevent medicine, a deterioration of aging, and this is something that has to be done.
So that's a good answer, but I want to tell you, if you listen carefully to your doctor, and if you see that there are signs of aging, then get a younger one.
Okay, and I think that
attenzione che parlo di professore di endocrinologia ordinario a livello internazionale,
yes.
non uno qualunque, attenzione.
Yeah. Okay.
Forse non so se è un aggravante.
Can you make a
If I make a comment, but this is really important. The science of aging is young and need to grow. There's no doubt about that. But we need to stop splitting the body in pieces. We split the body in the heart, the brain, the kidney, the endocrinologist, and the body is one. And until we recognize that all our parts are connected, and we need to have intervention and consideration about our body that are interconnected, and not just splitting the pieces and knowing about one part and not considering the other, we will not make progress in our aging process.
Thank you so much.
So.
That is totally right. And this is the new science. Science first was holistic, then it became very specialized, and now we are going towards somebody, a friend of mine, a great scientist, say Leonardo da Vinci, and here in Milano is the city of Leonardo da Vinci. Leonardo da Vinci way of looking at global problems. And so this is something very true. I'm very happy that this was so lovely and our public participated so much. I think it's a great success. So you'll have a chance to listen to our speakers if you stay today and tomorrow. And now we have the first scientific panel. Not that this was not scientific, but this was more like a talk. I would like to call Thomas Paloschi. He's a young doctor, work as a... Thank you. Thomas worked as a... Oh. So we even have music to entertain you. I want to say that Thomas is a young longevity physician from Italy. And he works as a researcher at NYU in stem cell and aging. So number one, I will have a lot of young chairs today and tomorrow. I think it's great to not only hear the scientists, many of them are young, but also to have the new generation, because longevity and healthy aging is for the new generation. I wish it was for me too, but I think it's too late.
Thank
so
you.
Thomas, you're an expert on stem cells, and we have two great experts coming. And please, Valerio and Juan Carlos, come to the stage.
Thank you very much. And because I've been given the stage as a young physician, a longevity physician, I would like to ask you all to stand up, and I'll show you how the future of longevity conferences are going to be done, so that you can move around and get your hallmarks of aging at bay. And if we can get some music. flow going. And so, I would like to thank all these amazing scientists. And as a young physician, I have a lot of trouble with all the Wild West information, trying to get a focused, information to our patients. So thank you all for this. And today we'll be doing a tête-à-tête between Professor Orlando and Professor Belmonte. They're two remarkable scientists in the field of genetics and regenerative medicine. So I would like to welcome Professor Orlando first. He has worked in Italy at San Raffaele Hospital in Milan and Dulbecco Telethon Institute in Rome. In 2013, he extended his expertise at the King Abdullah University of Science and Technology, KAUST, in Saudi Arabia, where he leads now the Environmental Epigenetic Research Program. And he's an elected member of the European Molecular Biology Organization, and he's at the forefront research of how epigenetic influences longevity. So please welcome Professor Orlando, and he will be speaking about ancient RNAs to keep you young. Thank you.
So, morning everybody. It's great to have an opportunity to have a tête-à-tête with Juan Carlos. Never had it before. But in order not to make a tête-à-tête with Juan Carlos, this is going to be a let's say, concept-oriented talk. So I apologize with the colleagues in the front row and introduce some of the concepts that somehow are leading the research in longevity. I come from epigenetics, so epigenetics in the 30 years of career started but nobody was looking at it as an interest. Oh, no, it was interesting, but let's say not explaining many things. But at that time, our community wanted to be inclusive and called from everybody working in gene regulation, development, other aspects of fundamental biology, molecular biology, but we needed people from different fields, from plants, from yeast, from mouse, Drosophila, whatever, to try to explain the complexity of gene regulation that was not only limited to transcription factors, DNA code, but there was something else behind it. So I think my personal opinion is that, in fact, longevity field is now transitioning in a similar phase, where we need people from any other field to try to contribute to the understanding of a very complex phenomenon that goes way beyond our current schematic understanding of even health. So starting from introducing epigenetics in a way that most people are familiar with the concept of DNA code, and basically that DNA in our genetic program would explain most of our even destiny, whatever. But in fact, this is not the case for a number of reasons. First reason is twins, are genetically identical, but in fact, these are two different people. So how do we explain this diversity? Diversity comes from the fact that, in fact, our biological identity, beyond the fact that we look similar, comes from our biological experience. So this is something that builds along the way with life. And all these parameters that we see, nutrition, individual care, adaptation, stress, access to knowledge, and everything, these are all parameters that happens to be now very relevant also for longevity or aging and so on. So the complex of these components, bad or good, makes the difference. And by making the difference, this means that there is something about the DNA that makes a difference. This is how the DNA is actually organized in the nucleus, and we realize that, in fact, it's more than just DNA. There's a lot of components that talk the language of environment, which is, at the end of the day, metabolism, chemistry, a number of factors that actually change a little bit the functionality of DNA. So, in fact, if there's a kind of short movie was supposed to play by the... in fact, what we see is that DNA is organized in time and space, two important parameters in life also. And this time and space means that there are a lot of components that record chemically the biological experience and decide which genes have to be on and off in different tissues and to what extent they have to be regulated. And this ability to change a program from starting from a one genetic program actually change with time. So our ability to diversify this information and translate it into the cell language in a way in functionality changes with time. So there are a lot of different levels of organization of our genetic material that influences the quality of our life and how we can actually record it, build a memory of it, and use this memory in the rest of life. So RNA, now everybody up there is aware of what RNA is because of the COVID, plays a fundamental role. It's a key component that mediates many of these levels of organization of the genetic information. Now, the surprise came from the sequencing of the human genome where, we know that RNA, DNA, protein, whatever, but the surprise came that only 2% of our genetic material codes for proteins, so structural component, functional component. The rest is surprising RNA with diverse function. To make the thing even more complicated, 50% of our genome is actually composed by repetitive sequence that have an interesting origin in evolution that are also major RNA output. So clearly 50% of our genome needs to be understood in function. Now, the beauty of it is that in evolution, these are elements that are repeated tens of millions, hundreds of millions of times in a way, have a history. That's why they're ancient. They are viruses, pathogens that our ancestors experienced. And somehow these invaders have invaded our genome. Invaded our genome and kind of built a kind of a evolutionary memory of the environment in a way or another. So we have to, and these guys can multiply, okay? Now, by multiplying, Barbara McClintock deserved the Nobel Prize, the intuition, they contribute to diversity. Diversity is so important, but the question still is now whether they are parasites or benefactors, because the fact that they jump around, they create mutations. Mutations, oh, bad. But in fact, this is the open problem. Now, mutations can also, produce variation. You see that all in nature and so on, there's several that you can appreciate here. These are single or multiple changes in repeat distribution in the genome that make a difference. In grapes, for example, Pinot, Cabernet Sauvignon, others, there are, from a genetic standpoint, very similar, whatever, but the parameters, the same changes apparently are the mobile elements jumping around and so on. Now, recent works indicate that individual single insertions in the genome can make a difference even in the history of our species. And beautiful work from Joanna Wisoska shows how genes that control the face, for example, the craniofacial aspects and so on, they have differences in the insertion of these elements that then can influence how genes work and so on. Now, there is an activity, surprising, of these jumping genes, so to speak, also in the body. So that wasn't expected. It's the work of Fred Gage that anticipated this. So instead of being bad, these guys somehow are there also during normal life. So the question is, what are they good for? Now, an interesting concept then starts that they can be beneficial in rewiring gene networks and contribute to phenotype variation. In the evolutionary terms, this has to do with millions of years. But it could be that they could be important, and they are now becoming more and more important in the duration of our current lifetime. So a completely different time scale. The first one mediated by DNA. The second one mediated by RNA. So in fact, these RNAs, these ancient RNAs, play a very important role in two different compartments of the cell, the nucleus by interacting with the epigenome. The other one is in the cytoplasm. And the basis of innate immune response and inflammatory response is one of the hot topics in longevity. So talking about the nucleus, so understanding the role of these elements in participating in nuclear architecture seems to be an important thing to go for. To go for because the observation of mattering for longevity is that retrotransposons, jumping genes, actually are re-expressed also during aging. They become de-repressed. So they could somehow create problems because of the jumping ability. Now, the question is that what are they bad for or good for? And in particular, what could be the component, RNA component, and next to the epigenome aspects. So we had the opportunity to think about it together with Juan Carlos, and compare our ideas about this. So I approached Juan Carlos and proposed a collaboration years ago, using his model system that he will talk about later on, progeria. So this is a genetic example of accelerated aging, and ask the question, what is the connection with the retrotransposon derepression? In particular, who comes first? Here, the progeria has a problem with nuclear architecture, epigenomic function, and so on, and then consequently, retrotransposon reactivation. But our question was, who comes first? Is this retrotransposon important, and so on? In fact, a series of experiments show that in the retrotransposon derepression, the jumping, so to speak, RNA, whatever, comes first. And then this initiates the process. So we thought that targeting this RNA could have been beneficial. And this is the part of the results, where you see that by targeting in vivo, the line 1 type of RNA in particular, you can tune it down, and then this led to very efficient tissue repair, genetic program repair, and lifespan extension, and another series of important parameters, including the so-called epigenetic clock resetting. So very, very efficient, apparently. Now, this is the conclusion of the story that we published, and so on. Now, this indicator wrote that probably targeting retrotransposon RNA, line 1 is one, but there are a few others that are in the list, and so on. And now the technology is in the hands of Altos Labs. We hope that it will be extensively studied. Second part is we go into the cytoplasm, another concept. So frailty, resilience, with age, we heard of that. We also lose performance somehow, the ability to cope with the environment. Again, here, the importance of how we adapt the genetic program through the epigenetic regulation to deal with the normal environment and time passing. So, and inflammation is actually at the center of this, okay. Now, inflammation, innate immune response, is where retrotransposons, these repeat components, are at the center of the stage. And this has to do with our evolutionary history, because they come from pathogens, so invaders, that now the cell has integrated into our genome, and then use this as a recognition for when similar invaders come, and and fight them using the immune system. But the new immune system is not only good for curing disease, but also to repair tissues and favor, for example, tissue regeneration. So again, here the question is, what are they good for in this particular concept? The ability of tissues, for example, to be repaired or supported. So inflammation has a good side or a bad side. So indication there in the clinics, but also in the experimental science, that controlled inflammation is actually key for tissue regeneration. So when you break bone or skin and so on, you need actually inflammation, but this has to be transient. If this is not controlled, tightly controlled, you go into the chronic inflammation, and then basically things go wrong. This is particularly relevant in aging, because the chronic inflammation starts to take over. So there's something that loses control at the level of these repeats. So for a number of reasons, we then looked into osteoporosis. Osteoporosis made us curious, because we actually observed that in osteoporosis, where you have a defect in frailty on the bone, the activity of retrotransposons was reduced, severely reduced, strongly reduced. So we thought then, instead of getting rid of them, by eliminating them, to provide now stem cells that are mesenchymal stem cells with line 1 RNA. So an experiment did before COVID, so something a little bit visionary. So delivering the RNA. So this is the measurement, in fact, of the fact that osteoporotic material, which is a collaboration with the hospital in Zurich, we have donors of this. The amount of the activity of this line is very reduced, and this is actually tissue specific for the bone. So by delivering the RNA to mesenchymal stem cells of the donors, we restore pretty strikingly the bone formation, or say mineralization production from the cells. This was very striking, and if you can provide other types of RNA, nothing happens. So this is something that has to do with the quality of the RNA that has particular structures and so on. Now this is, I don't have time to go into the details, but it's getting more and more interesting because this involves a signaling between different cell types that initiating from the founder cells like mesenchymal stem cells, triggered with RNA, this Asian RNA, start to secrete factors that then instruct other cells to start the bone regeneration. And these factors, we have characterized them. They include a variety of different components, but they are very, very specific that seem to be the job. So this is a case of a good side of, again, good side of retrotransposer. Similar concept works in other pathological or age-related concepts like this is a muscle dystrophy or high-fat diet, for example, is Nash Laffold, where there are interesting connections with that. Now we also compare the bad high-fat diet or the good fat like olive oil, and olive oil actually is very good because it kills the bad effects of palmitic acid, for example, that triggers chronic inflammation through repeats, but at the same time does not elicit any response from the repeats. So there is an interesting component here. So Asian, to end here, is good somehow. So we got good information from very, very old, even invaders of our genome, bad guys, viruses and pathogens and so on, that allows us now to deal in a more efficient way with environments. So a question now, I think it's a question also for later discussion, is whether with the current knowledge, is it a matter of going back in time by erasing the memories of rejuvenation in a way, in resetting the epigenome and so on, or should we protect the memory for a better time? That's an open question. Okay, it's done. Okay.
You can sit over there if you want. Perfect, thank you. So next, we're honored to welcome Professor Juan Carlos Izpisua-Belmonte. He's a prominent figure in biology and regenerative medicine. He has a strong academic background and worked in Bologna and has studied in Bologna and University of Valencia, and over 30 years of experience, including a notable period at the Salk Institute. And Dr. Belmonte now is the founding scientist and director of the San Diego Institute of Science at Altos Labs. So he's recognized by the Time magazine in 2018 as one of the 50 most prominent and influential people in healthcare. So please welcome to the stage Professor Belmonte. Thank you.
Thank you, Thomas, for your nice presentation. Thank you, Viviana, and all the organizing committee for this wonderful conference. On the left, you see how we all here start our life. It's just the encounter of the biggest cell of our body, the oocyte, and the smaller cell of our body, the spermatozoid. In a few days, you start to see a ball of cells. At the bottom, you see pink cells. These are the so-called stem cells that will differentiate into the more than 250 cell types that constitute our body. And in about 5 to 6 weeks, all these cells start to get spatial organization that leads to this embryo you see there. And finally, after 9 months, a baby is born. For the first, I would say, 30 to 40 years, disease is not something that we occupy too much time with. But after 40, 45 years, disease starts to appear in our life. And it occupies a big part of it. In the last few years, we have started to map at the cellular and subcellular level this process of our life trajectory. And this is a nice image I took from Klaus Rajewski's lab, where you see a trajectory of healthy cells in blue. But at some time during our life, this healthy trajectory of cells start to deviate and make other colors, as you can see here. And the question that many of us are working here and many of you are interested on is try to see whether these disease trajectories that deviate from the blue trajectory can be reverted. If a disease trajectory can be reverted in our life to a healthy trajectory. Perhaps the answer to this question came from this very important experiment that Dr. Shinya Yamanaka performed about 15 years ago. He took an old cell, an adult cell, that has already gone through a specific trajectory in life, add 4 transcription factor, and brought these cells back to the very beginning of the life, to embryonic-like cells. So he was able to reverse the trajectory of an adult cell into an embryonic-like cell. To me, this is perhaps one of the most important experiments that have been done in biology. How is it possible that he just take a single hair and make a mouse with it? This is really incredible. How does it work? It's now 15 years since he published this paper, and still we don't know very much how it works. One component of how this takes place, it was presented by Valerio, the importance of the epigenome in this experiment. As Valerio indicated, there are 2 types of chromatin states in our cells, open and closed chromatin, euchromatin, and heterochromatin. Now, when we think about aging, in many occasions we think about something that has gone wrong. There is some damage. Our mitochondria are damaged, our DNA is damaged, etc., etc. Another way of thinking is that our epigenome has changed. This equilibrium between open and closed chromatin that we see in a young cell maybe is altered in an old cell. And in fact, this seems to be the case. This is an oversimplification, but you can see on the right that a young and healthy epigenome has more of this closed chromatin versus the old and a disease epigenome that has more open chromatin. This is a generalization, I know, but just to indicate that there is a correlation with the structure of the chromatin, disease, and aging. Now, what Shinya was doing by adding these 4 factors to an old cell was resetting the entire epigenome of the cell. And by that way, he was able to get to an embryonic-like cell. What we did a few years later, and using this mutation that Valerio has indicated, this progeria mutation that in mice is very similar to a human. There is an accelerated aging process. You can see in this mouse, it has less hair, is more fragile. By adding now Yamanaka factors, but in this case there was a small switch. Rather than having the factors all the time, we are just applying them in short pulses. In this occasion, twice a week. And the effect was not going back all the way back to an embryonic stem cell. It was a very different effect. We could see that that mouse still was a mouse, was not an embryonic mouse. It was an adult mouse, but it looked very different. It was younger and it lived longer. Now, if we look at the epigenome of this mouse, you can see here in these panels, this is a particular epigenetic mark that was not present in the progeria mouse, but after expressing these factors, then that particular epigenetic mark went up. But more important than this, just changes in the epigenome, is the other two panels. Here in the middle, you see that the nuclear envelope of this progeria mouse, they are distorted. They are not normally round how it should be. But after the epigenome is changed, you can see that the nuclear envelope is now normal, that the DNA damage is normal. So this is very difficult for me to understand yet. We change the epigenome, and then the damage that is in this cell get fixed. How is this happening? So I tend to think that we're cheating the cell to believe that has a young epigenome, and the rest happen automatically by itself. What does it mean in terms of disease? So I'm telling you that, and this is the best example of the explanation I'm having. If we were to take these mice and we fix the damage, which is a mutation, we take a CRISPR technology, the mouse now doesn't have the damage anymore, and it can live 18 weeks longer. However, if we do the same experiment, we don't fix the mutation, we just change the epigenome, the mouse lives 18 weeks longer. Still, it has the mutation. So somehow the epigenome has been able to compensate for that mutation, to buffer for that mutation, and repair in a way that I still don't understand the damage that this mouse has. So it's telling the cell, you just get younger, and then you do whatever you have to do. And that goes back to my first slide. For the first few years of our life, disease is mostly not present because the cells are younger. It's when the cells get older when disease starts to appear. If you were able to rejuvenate the cell, maybe disease will take longer to appear. And this is a clear example of this. But rather than focusing in aging, what I would like to go back is to a question that was mentioned this morning. Aging is probably one of the biggest disease risk factors for diseases. Here you can see that around 40, 45, 50, all these diseases, any disease, start to increase logarithmically in the human population. So to try to see if this partial reprogramming, these short pulses of Yamanaka factors, had an impact on the disease, we have generated many mice where we can express these factors in a specific organ and tissues of the mouse. And I'm going to give you a few examples of how these short pulses of Yamanaka factors for a couple of days is able to improve many diseases. For instance, here you have another mutation, this leptin mutation. The leptin mice, they eat a lot. They cannot stop eating, and as a consequence, they get fat, here on the left. If you give a short pulse of these factors, they keep eating, but their fat is less. A similar phenomenon occurs in a high-fat diet. This is a normal mouse that was given a lot of bad food. It gets obese. But if you, at the same time, you give a couple of cycles of these Yamanaka factors, and you keep giving the food, the mouse doesn't get fat. You can see the difference in the fat content in this on the right. There was a discussion this morning about this Ozempic drug. You take the drug and you reduce your fat, but you also reduce your muscle. And if you do that many times, probably you will get sarcopenia. Now, if you take the same 4 factors, a couple of cycles, you can see here just by looking at the 2 images, this is an injury that makes this mouse muscle a little bit more fragile. After you give a couple of cycles of the Yamanaka factors, you can increase the muscle strength. You alter the differentiation of muscle stem cell, and you increase the capacity of these mice to be stronger. So You don't need to correlate always losing fat with losing muscle mass. Another example is, for instance, a toxin. Let's take the case of a drug that we take oftenly, acetaminophen. So if you give mice a high dose of acetaminophen, that's not going to be good for the liver. In fact, if the dose is too high, about 10 days after of giving a high dose of acetaminophen to a mouse, 100% of the mice will die of acute liver disease. If you add just a couple of cycles of these Yamanaka factors, you can see on the red line that 50% of the mice, they don't care about this toxin. They are able to survive and deal with the toxicity effect of this drug. And just for instance, another example, here you can see the function of aged mice following these short cycles of Yamanaka factors, how the different tests that we have in the lab to assess the brain function of the mice, they all get improved. It was mentioned fibrosis and inflammation. If we have done an analysis of what is the major pathway that changes with aging. The major pathway that changes with aging is epithelial to mesenchymal transitions, which are at the very core of inflammation and fibrosis. Here, in the case of fibrosis in the liver, indicated in red, after a short pulse of the Yamanaka factors, most of the fibrosis of the liver disappear. Now, I could be another hour telling you examples of how, when we apply these short pulses of factors, improve many aspects of the disease progression of the different mouse models that we have created. But there was a question this morning that indicated, well, your particular molecule, how is it possible that can alter a complex process like aging? That's difficult to understand. And what I'm telling you here is just one particular treatment. In every situation that we have put it, it seems to improve age and the disease of these mice. If we look at the way medicine is done today, it's a very highly specific medicine. If you have a mutation in your DNA, you could opt for doing gene therapy. If you have an altered molecular function, either in protein, in metabolism, etc., one can think of developing traditional small compounds, traditional drugs that can target this altered function. You could also think, and thanks mainly to the studies of Dr. Shinya Yamanaka, bringing cells back to an embryonic stage and differentiating them to the cell type that you want, introduce them through cell therapy. But what I'm telling you is something very different. This is not specific. I put it in a liver disease and seem to improve the disease of the liver. I put it in a muscle problem and seems to improve muscle. How do we interpret this? Valerio introduced this image here where we can see on the left and on the Y and X axis how in a young cell, in a young organism, after a stress, there is a response. And in an old organism, a fragile cell, you can see that these two lines, they meet at a very different space of this diagram. What we want to indicate with this is that the cell, while it's young, is able to be more resilient and more responsive to many different types of stresses. When the cell gets older, and I would like to think when the epigenome of that cell changes, then the response is less efficient. Cells lose their identity, and therefore they cannot cope as good as they were coping with injury and disease while they were young. So if we're able to cheat the cell until you are younger by modifying the epigenome, the cell will take care of the rest. And this mouse that I have shown you that has a problem, specific problem, a mutation, a DNA damage, an alteration in the nuclear envelope, just by changing its epigenome is a good example of what I have told you. Now, Viviana was saying this morning at the end, all these treatments seems to work in mice, and we should be very careful that you cannot always translate what you see in a species to a human species. And I totally agree, but we need to start translating all these different treatments. And one of the examples we have started to try to translate these particular interventions has to do with the shortage of organ for transplantation. About every 30 seconds, a person dies in the world because an organ cannot be found for transplantation. So we decided to start doing an experiment, and the problem is the following. How do we specifically deliver our molecule to an organ? If we put it in the blood, it will go everywhere. And could be good, as I have shown you for the mouse, but we don't know the possible toxic effects that this treatment could have as well. If we were to localize our treatment to the cell type or tissue that has a problem, maybe the toxicity effects will be alleviated somehow. In the last few years, engineers and doctors have come with machines like this that can keep our organs before transplantation for a few days, a couple of days. And that's really incredible because it allows you a window where you can alter somehow that human organ before transplantation. In the last couple of years, we have been working with a very talented group of clinicians at the hospital clinic in Barcelona, and we can maintain now a liver for about 14 days in the perfusion machine before it's transplanted. And that's really a window of opportunity for research, for scientists to try to improve that organ. A big percentage of the organs that are donated by a very generous person for transplantation doesn't get transplanted. The doctor looks at the organ and thinks that the quality of that organ is not good enough for transplantation, and it just gets discarded. So if we were able to maintain for some days that organ that is going to be trashed because the doctor doesn't think that the quality is good enough, and trying to improve with any of our treatments, this certainly will be a major change in the field of organ transplantation. So we have started to do this, and here you can see that if we take a virus to deliver our Yamanaka factors to a rat, in this case on the right, it doesn't work, so just look at the right. We inject in the pelvic area, you see the injection. Most of the virus just get stuck in the liver. So if we were interested to bring the treatment to the kidney, nothing will go there. The liver somehow is the organ that absorbs most of the things that go through your blood. Now, if we do this perfusion, which is we take the rat kidney, we put it in the perfusion machine, we deliver in the perfusion machine our vector. You can now see here, and transplant it back to the rat, that the color is only in the kidney, it's not in the liver. It's a way to avoid that the treatment goes to the liver, but just stay to the kidney. And so we have done this experiment in rat, and what we have taken is an old kidney, a kidney that is already fibrotic and is not working that well. And we have put it in a young rat. As a consequence, that rat is going to live less because the kidney that we started in the process of transplantation was not that good. However, if at the same time, that will be the control, we treat that kidney with a short pulse of the Yamanaka factors while in the perfusion machine, and then we transplant it to the young rat, eliminated the 2 kidneys from the rat, so the rat now has only the transplanted kidney that has been treated shortly with these factors. You can clearly see on the right that there is a big difference in the survival of that rat. The rat with the old kidney, in blue at the bottom, lives less. They start to die at a particular time point. While the rat with the old kidney that has been treated just for 30 minutes with these factors, it can live much longer. So this, I feel, could be a very good first example to start translating our results that we see in an animal model in the lab with a human organ that is discarded. It can be treated for a few days in the perfusion machine. If after a few days the doctor realizes, oh, this organ now that I was not going to transplant, that I was going to trust, can be now used. Maybe they change the life of that person that was expecting the organ may change. And it will be maybe a way to demonstrate that all these treatments that we use in animal can be translated to human. This is just the summary of what I was telling you. And I would like to, especially because Thomas is telling me that we're done, everyone that has been involved in some of the data that I have been told you. Thank you very much for your attention.
Would you want to sit? Thank you. Great, thank you so much. So just to set the stage a bit, if you don't know what Altos Labs and KAUST is, they're 2 very important labs, and there's, I think, around $20 billion involved in the KAUST Institute, and there's around $2 billion are at disposal for the Altos Labs. So is it possible for you guys to give the mission of the labs and explain what you're doing within?
You start?
Okay. Briefly, I think the mission of Altos Lab is aligned very well with the concepts that I have tried to explain today, which is the idea that if we can increase the buffer capacity, whatever that is, that we don't know yet, of an old cell to the levels of a young cell, the young cells somehow will be able then to deal with all the problems, with all the injuries, with all the diseases that one way or another we're going to be faced. There are different ways to do this, and there are different ways to measure this. One of our colleagues, Steve Horvath, probably will tell you a little bit more, but the idea is that rather than being very specific about how to treat a particular disease, try to understand, it could be the epigenome, it could be the mitochondrial function, the energy of the cell that somehow with time it's deteriorated, and by bringing it back to a younger cell, try to improve disease. It is composed of many basic scientists that we have brought from the academia with different interests and different angles to attack this problem of how disease appears and how we eventually could reverse by this concept of increasing their buffering capacity, increasing the youthness. That will be, in a couple of words, what our mission is at Altos.
Perfect.
Our mission, impossible, by definition, is, maybe changing a little bit of the perspective or the standpoint. In order to understand disease, or anything that is linked to disease or leads to disease, probably we have to go back to a better understanding what health is about.
I mean, the deviation concept that Juan Carlos illustrated when things start to go wrong. Still today, we don't know very well where is the junction, where is the divergence point. Whatever AI is there, but I think that we need data. We need a lot of, making things a little more complete for that matter. So, in my lab, the idea is to go back to a better understanding of not the bad side of things, but the good side of things. The retrotransposons, repeat elements, this source of troubles, or junk DNA was called for a long time, useless. 50% of our genome defined useless, okay? It's there for some reason. The fact that now we have an understanding with the fundamental aspect of cell physiology, which is epigenome regulation, so the ability of cells to cope with the naturally changing environment and adapt gene regulation. This is actually has these RNAs at the center of it. At the same time, the involvement of these RNAs in mediating innate immune response, so inflammation, which is then understanding the good side of inflammation. Probably this will, and understanding that the source of it, the controls of it, may help us to find the keys to modulate positively inflammation and then maybe have beneficial effects on rejuvenation, tissue regeneration, or whatever. Keeping in mind that these are very complex phenomena that need to, improve our complete understanding how things actually are regulated at multiple levels.
Perfect, thank you. So I wanted to maybe give the audience and physicians the ability to see how these scientific discoveries are going to be implemented in the clinical practice and how people can use them today, or what is the timeline for them to be accessible to the public?
This is a very difficult question. I feel that there is exciting discoveries in the animal models that we're working with. The process of translation and translability of these results in an animal model to a human usually takes many, many years, and there are many roadblocks that appear in the process. Therefore, to venture a particular time for this treatment is certainly difficult. We start to see examples, and they were discussed this morning, with particular molecules. We were talking about rapamycin, for instance. There are studies that have been already translated to the human, to monkeys, to many other species, and clearly indicate that the path for this could be shorter when properly done. There are other treatments like the one I just mentioned, where trying to even understand how the epigenome of these cells that have been treated with the Yamanaka factors changes. So it's very difficult that if we don't even understand how things work, that we can translate this in a short time. So the answer is, I don't know, and I could have done it much earlier. Just say, I don't know. Okay.
Thanks. Probably the point is how much time it will be needed to change the culture, so the way we look at the problem. And probably this is the transition time. I mean, genetics taught us to look at phenotypes, defects, by looking at the gene and then a resulting phenotype. But this is the monogenetic diseases, which is the very, very small percentage of them. Now, if we forget about disease and look at, explain the phenotype, so how a person is about. Nowadays, if we go from DNA, RNA, so we can say, well, what is the transcription program of the cell? Then we have metabolomics, so the metabolism of it, the resulting. So current approach is a multi-omics approach. So we need to stacker all these levels of proteomics, RNA, DNA, metabolomics, putting them together and then make sense out of them. So this already changes the way we look at the phenomena, which is already very complex, of a cell. Okay. Now, if you look at aging, now with this type of parameters combined together, it probably is a multidimensional way of looking at this complex phenomena. the same approach has to be used to look at different, more classical type of problem, like developmental biology. I mean, explaining development, how decisions are maintained, kept and maintained in very complex organisms and so on, is still, something we have not figured out. And, Juan Carlos comes from exactly this type of mentality. The fact that now we have this magic 4 factors that can reproduce something, but this is a kind of approach that seems to simplify things, but as Juan Carlos said, we still have no understanding how certain things work. So now we have new tools that probably, if I may, will change also the way, the type of drugs we are going to develop, having an understanding of which phenomena, which cellular pathways we are interfering with, and this which will translate in a completely different medicine.
Thank you.
So I want to add, yes, more of what you said, Valerio, and that relates to the question that I think is the most important question that I heard this morning is, how is it possible that with the complexity of the phenomenon of aging, you dream that just by altering one particular pathway, you're going to change everything? You need to approach the problem from different angles, and this conference is a very good example of this. There are speakers that are working in the epigenome. There are speakers that are working in how protein folding and degradation take place, and we will hear this from Ana Maria. There are speakers of how mitochondrial and energy play a role in this process, and until we don't have a way to integrate all these different angles, it will be very difficult for us to understand what do we mean by an aging cell. It's a very complex problem. At the same time, we have to be optimistic, and I really appreciate Viviana's organization, because your question couldn't be better addressed by having all these different angles at the same problem.
I have a different question, but Valerio is Italian, you are Spanish. You work in the States, you work in Saudi Arabia, where a lot of money is invested. Altos Lab, I think, started with 3 billion, if I'm not wrong. You have a fund, very new fund in Saudi Arabia called Evolution, 120 billions, if I'm not wrong. Is there a hope for a country in Europe, we don't have this amount of money, to have to get back, some of our scientists who are going abroad, because that's where you can do proper research, well-funded?
But I think the amount of money that one institution, one foundation is investing alone will not make a difference. I think probably, especially for longevity, the point is to create a network, create a culture that involves more subjects, more countries, more, hospitals, more academic context, more industries, and so on. This will mark the difference. The commitment from individual bodies to go in this direction is, of course, the first condition. But that's why an event like this or others can contribute to change the culture. In Saudi Arabia, I belong to an organization like CAUS. CAUS was created less than 15 years ago with a mission. Which a mission was to change the social context by education. That was the mission. No products, no industrialization, whatever. Educating people, starting from education. And contributing to the world, saying, we want to understand the climate, we want to understand energy, we want to understand biology, medicine, and this and that. And producing a new generation of young people that can help us to solve other problems in the future. Social revolution. Then came evolution, which which is much more recently, which was launched two years ago, with a decent amount of money again. But their vision is, again, global. So they put a lot of money and they invest globally, not only in Saudi Arabia, with the idea that if we get together, we join the efforts, then we probably mark a difference, and maybe we accelerate certain processes and so on. So the hope is that individual countries will then participate in this process and then favor also the mobility of people that decide to be behind this mission.
That's a very good question, and I would like to add on your point about education. We have been educated about the importance of trying to eliminate cancer in many, many different ways during the last 30 years. Not just in Saudi Arabia, not in the States, but in Europe. And that education in the street has led to our administrators, our politicians, to put resources into the problem of cancer. Even though some statistics say that if we were to have the magic pill to cure all cancers, we will live just about 2 or 3 years longer. The idea that cancer is a disease that needs to be attacked and resources need to be deployed, I think is all over the world. You are asking how we come from Spain, from Italy, how Europe could somehow dedicate more resources to, as I said, the major risk factor for any disease, including cancer, but for any disease. And I feel it's education in the regular people to realize that this affects our life, that it's not just a particular disease, but is the major risk factor, and in the end, one can increase health span by studying this problem. Education is my answer to your question.
Right. Actually, if I may, the paradigm shift will be, again, to take a decision whether, quoting Mario Draghi, whatever it takes. You know. So the point is whether innovation, nobody can predict where it comes from. The point is not to accelerate, force, processes, but to amplify the possibilities, the chances that things happen. I mean, if you look simply to the most recent Nobel Prizes, I mean, RNAi were people that were looking at, C. elegans or fungi, genetics, and whatever, nobody. CRISPR-Cas9, bacterial recombination, or others, and so on. The COVID vaccine and so on. People that, behind it, that at some point had even problems in receiving funding or even getting a job, So there's no way, I mean, the decision, the strategic decision is to invest in education in the sense, give back to people and the society the fundamental right of knowledge. And in a way, give people back this fundamental right will be an investment which, will pay off by definition. In the past, you mentioned cancer, but for example, the example of neurodegenerative diseases. Industries put a huge amount of money in this area and created, institutes, whatever, and so on. And after decades, not a single concrete solution has been found, probably because of the approach. Maybe the approach was the wrong one, so the way of looking at the problem was not the correct one. So the shift will be if we start looking at things in a different way, with the proper technologies, we will increase the chances to find concrete solutions. So it's a cultural.
Yeah, makes sense. So anyone in the public want to ask a question, please take the microphone.
Thank you very much for that talk. This question is for Professor Belmonte, and I'm not a scientist, so I'm sorry if I am not asking this clearly enough. but I'm curious, you were talking about the cells and how they become damaged over time, and I just want to understand, how is it possible that cells accrue damage if they die and then replace themselves? I'm just trying to understand how that damage is accrued over time, and also if it's possible that it's something about the cell environment that is damaging the cells.
Your question is well formulated, and you don't need to be a scientist, but you are very well educated the way you formulated the question. And that's why I say the importance of education in the street in a person who is not a scientist. You asked for two questions. There are cells in our body that live for a long time. Many of our brain cells, most of our cardiomyocytes, they live for a long time. So there is time for them to be exposed to insults and be damaged. Not all the cells get replenished immediately. The second question is about the environment. Yes, it is very well described that the environment may affect the function of a cell. There are cells that enter in what we call senescence phase, which they start to undergo an unhealthy process. They secrete molecules in the environment, in their environment, that affect other cells that is going to be deleterious for this function of these cells. So this is very well described in the literature. In fact, as we age, the proportion of senescent cells that secrete these bad molecules for the environment increase with time.
If I may add a comment, thanks to the development of technologies now that can go down to the single cell level, we are now understanding that even in each organ there exist different cell types, or even say similar cell types, but they don't actually are exactly the same. So the point now is to understand how these cell types talk to each other. So it's a complexity within the complexity, and this is called spatial omics, so-called. So you can go from the histological prep that is used normally in clinical diagnosis and so on to the single cell level and create multi-omics type of data that help to understand what is going on at the single cell level in a given organ. So this means a transition in our way we look at data, we produce data and look at data and maybe make sense out of them. So I think this is a frontier that's going to produce a lot of interesting advancement, because when you then go to, for example, deliver a drug or a concept, whatever, to an organ, then you have the possibility to look in high details to what happens to each of the individual cells and make a better prediction.
Thank you both.
You need a mic.
I have a question from our online guests in Italian. Considerato che dovremo attendere ancora molti anni per avere risposte dalla ricerca scientifica, vorrei un parere sui fattori ambientali che possiamo modificare subito, come la nutrizione, l'esercizio fisico, il consumo di integratori, lo stile di vita sano. Secondo voi, in che misura possono determinare cambiamenti genetici ereditari? In che misura potrebbero determinare un miglioramento della longevità umana? Grazie.
Epigenetics.
That's a good starting point. I would like to highlight and advise you to come, I feel it's tomorrow, or if not, the day after tomorrow, the talk of my colleague Steve Horvath. He will present to you, I hope, maybe I'm just advancing this, how changes in diet, changing in exercise, it can change your aging clock. So very basic activities. Many other interventions that change and alter and make younger our aging clocks. All these treatments that we are trying to accomplish still are somehow far away, we don't understand. But the very simple activities like moving, running a little bit, eating a little bit better, they have a big influence in this epigenome that I was mentioning before that can rejuvenate your cell?
If I understood the question well, there's also an implication whether any improvement now can be transmitted to the next generations via, reproduction. And this is a hot topic. To what extent our biological experience can be transferred to our, future generations? I think it's a relevant point. Well, this is something that, technically speaking, the biology in humans and other models and so on seems to start to understand. Something can happen. Okay, there are certain habits that can be somehow transmitted and so on. But at the same time, we have to understand what is the duration of this and what is the significance of this, whether this is a real advantage or disadvantage. If stress is something that people learn and then inherited, or something else, even culture can be inherited.
We have one more question.
I have something interesting to say about this, because we had the other day a beautiful conference on centenarians and blue zones, and you all know about blue zones. And one of the things that it was highlighted by the scientist Graziella Caselli, who is the one who discovered the blue zones. And you know why they are called blue zones? Because they were using a blue pen to mark down each place where there were centenarians. And by the time they had a place with many centenarians, it became deep blue, and they were called blue zones. And one of the things they found is that in these blue zones, a certain number of factors like eating, exercising, going to sleep with the sun, coming down and waking up early in the morning, being in a social environment where everybody could talk, making music. These were all factors that made people healthy. And another factor was the mothers, and how the mothers transmitted to the young people the way of cooking, the way of being, the respect for elderly, socialization. Very interesting, there is in Okinawa, a blue zone in Japan, and many of the people from there who in the past had lived 100 years for generation in a healthy way, as soon as they moved out and they started eating bad food, being alone, being in a different environment, being in pollution and not moving, they started aging very rapidly. So I think this is...
Okay, I'm sorry for the person online who asked this question. I misunderstood, but I'm very happy you just translated for me. It was about a year ago that this phenomenon of transgenerational inheritance, we demonstrated that it takes place in mammals. We know this very well from plants. We know this very well from flies, from nematodes, and there is many evidences of this blue zone in mammals, so to speak. But it was very difficult to demonstrate whether this phenomenon of the inheritance of the epigenome into the next generation happened. We were able to take a particular phenotype, in this case obesity, and we have shown, and we are now by the 12th generation, where this phenomenon took place in one mouse, and it can be transmitted to the next generation, the next one, and the next one. We're now in the 12th generation of this mouse, and just whatever the mother was exposed can be transmitted in a particular loci. So certainly the way we behave is the message, is going to affect our children and the children of our children, the importance of the environment and the epigenome.
So there are more arguments for the psychotherapist then.
True.
More like your biology documentation of why your mother was bad. Anyway, blame the
So we have one more question to wrap it up.
Hello, my name is Thomas Streimelweger. I come from Vienna, Austria. I just wanted to contribute to the technology aspect, and I think it's a little bit of a crossover. I have co-founded, basically spun off 2 companies out of academia, and we produce, based on iPSC stem cells, brain organoids, so we can replicate parts of the human brain in a dish, and we can apply substances. That's a totally new way of drug development, and you will see that we can make a big jump forward beyond animal trials. So all these mice experiments will soon be replaced by real organ stuff out of iPSC stem cells. We have co-founded a second company that does the same with heart, so we can replicate the left chamber, a right chamber. We can fuse it, and it's contracting its real muscle mass, so we'll also have some progress there. And the other interesting aspect is that we have received just recently very strong interest from KAUST. Our people have been visiting Pierre Magistretti at KAUST, and Saudi Arabia might be available to fund these projects much more heavily than any European institution. That's also a little bit a bitter aspect of it.
Great. All right, so we'll close now, and I think there's a break, lunch break, and we'll see you after then. Thank you. Thank you very much.